it would be time to kind of turn it over to our pre-scheduled unanswered chat.
Yeah. Just 1 second. So I'm guessing you were showing how to search by DNA.
Yeah, kind of by
No,
provisional names specifically.
Okay, I'm gonna make our speaker. Oh, by provisional names. That's cool. I actually don't know that one. I'm gonna make our speaker, who is here, Alfredo Justo, a co-host, so that he's ready to present when we get there. And I've also got a couple of other people being co-hosts. As co-hosts, you'll see other people coming into the meeting room, and you could let them in, please, so that people can join us. Because I'm going to start sharing my screen to do the mystery mushroom presentation, and then I can't do it. All right.
Yeah, this is something we kind of thought up just a couple days ago, so we didn't give people a lot of notice, but
No, but I think maybe this will be our showing what we mean by this, and people can send in their mysteries, or maybe they came with their mystery mushroom links to this meeting. We definitely don't have to do just the ones I put in the presentation.
yeah, if
But
anyone did come prepared with links, go ahead and throw them in the chat, and I'll take a look. And
yeah, we can
if you
have
want us
some
to look
extra.
at something,
Yeah, please feel free to interrupt me so that we can check some of those out. So all of these today are coming to us courtesy of our chief mycologist of the Wisconsin Mycological Society, Tavis Lynch, because these mushroom mysteries are all coming off of a list that he recently sent to us here at the University of Wisconsin-La Crosse to be doing some sequencing for WMS. So this is basically, this particular mushroom mystery set is going to be your last chance to get it right before the DNA tells all, or tells us a good lead. This is a first example where it's a mystery, but Tavis did update it with a specific species, and I thought that would be a good one to start with because I was really curious what made you upgrade it to Amanita vulvata on your ID, Tavis.
it's either vulvata or pseudovulvata, and the descriptions are so similar. Pseudovulvata turns out to be more common than vulvata, and I don't know. I found this one a couple of years ago. I don't remember exactly why I decided it was that, but I wanted to attach one or the other to it. And maybe pseudovulvata was not recognized by INAT. Maybe that's why I did that.
Sure, so you were like, oh, well, if they're lumping them anyway, I might as well put this name
Right.
on
I
it.
know. I know it's an Amadella. I've got a handful, and actually, those plates that I sent you, there was only like 3 mystery mushrooms in there.
Yeah, I've pulled a couple extras out from
Okay, cool.
just
Yeah,
from
very
their
cool.
talk, because we're gonna also do some Pluteus at the end. So I've also got one just to prime everybody up. The last one is going to be a Pluteus, a deer mushroom, where Tavis and our speaker, Alfredo Justo, disagree. Because I've
Oh,
got to create the drama.
I love it.
Yeah. So this one is also really interesting to me as a Amanita where the cap is shaggy. Like I feel like I wouldn't immediately, those warts don't feel like regular warts to me, but they are, I guess, fragments. And I was curious if that was also a feature that you found diagnostic or no.
With subgenus Amadella, yes.
Okay, that answers that for me. Cool. Well, this one was just to get us started. It's less of a mystery because we did, I think, have a decent name on it. But assuming that subvulvata is a valid species concept,
Pseudo.
we'll be pseudovulvata is a valid species concept, then maybe we will find out more about it. Could definitely see why they called it vulvata or pseudovulvata because it's got a very large partial veil going on
You know
there.
what? I do remember this was a very small mushroom. So one of
Really?
the descriptive differences was, I believe it was cap width versus stem length, but this thing was only like 3 inches tall.
Right. And it looks almost the same wide as it is tall.
Right.
Very square Amanita. Okay, so first mystery put to bed. This one's probably harder. I saw you had it at family level, so higher than genus. We're not as sure about this one. It's something in the Quaternariaceae. Of course, they broke up that genus into a family with a lot more in it. or split up what used to be just Cortinarius and added more to the family. But any comments on this one for how we would, or like what's our closest thoughts?
Man, yeah, I'm gonna chime in right away. I mean, Cortinarius is so difficult. There's so many. A lot of the little brown ones we include in the subgenus Telemonia, and about half the Cortinarius are Telemonia. So, I mean, it's 50-50 shot if you just call it Telemonia.
Right, so that could be, is that one now a genus or a section?
It's a subgenus of Cortinarius still.
Subgenus.
Subgenus right now. Okay, so that's where we're at on this mystery. Any additional? I don't have any for this one particularly. That's why I was like, let's raise it. And people who are just meeting or joining us, you can unmute yourself to comment on any of these. Or put it in the chat and Brennan might read it aloud.
Do you want to grab one from the chat right away? We do have one.
Yes, I do want to switch to one from the chat so we can see it. Okay. So I'm going to go into here and get the link. Whoa. Oh, is it a picture in the chat?
There you go.
Okay. I'll download it and open it and then share my screen. Or I'm still sharing my screen?
You are.
Okay, great. Well, now y'all can see it large. So I
That's
saw
Clavaria delphus.
Clavaria delphus. Oops.
Yeah.
All right.
Which
so
one, Teophis?
Truncatus.
Truncatus. Okay, so I'm going to type it in the chat. Clavaria delphus, and then Tavis can tell me I spelled it wrong. Did you say so? Truncatus?
Yeah, that'd be my best guess. It's not a fairy fan whoever said that.
What
All
about
right.
Pistolaris?
Pardon me?
Pistolaris, is that a possibility?
I can go to my notes. Pistolaris, I don't know that we have that locally. We've got Ligula, Americanus, and Truncatus for sure.
Okay.
All right, somebody spell that one for me. Pistularis.
Pistolaris.
Okay, not one I'm seeing. Seems like I'm not spelling it right.
It's ILL.
Pill. And then you?
Pistill. Pistillaris.
Okay.
Is that like pistol on the flower anatomy? Like that?
Delphus pistillaris?
Yeah, spelled like the pistil on a plant. There it is. Oh.
Yep, and I'm checking the map.
Yeah, and actually go back and look at the morph on that one. It's not.
This one is not research grade, so that's not necessarily our guide there. Like at least 2 people have to agree for them to get to research grade. I'm going to pull back up our comparison pick here. And
I guess the color is off for Pistolaris too.
The shape too. I would look at Truncatus.
Okay, and this is where we're gonna.
That's gonna typically be flatter and wider at the top.
So, trying to go to the species page here, and I'm probably going to come up as a similar species, misidentified, edit filter by Wisconsin. Yep, so some people are the similar species tab is really useful to see what other people have swapped it for before. And we can look at the map on this one too. So both of them are here, according to iNaturalist at least. All right, so we can have our, let me see if I can do the fancy window splitting for you guys. We've got on the one hand, on the other hand, and in the middle. And this is, this is what I very often do. But it's not a substitution for going into reading like the description of the actual species. This is just getting a gut check based on, here's what people are putting as this name on iNaturalist, and here's what people are putting as this name on iNaturalist, which, we could all be wrong together.
Sure.
But I do honestly, Tavis, I don't like to disagree, but it seems like not everybody's calling, but a lot of these at the top here of truncatus have this very flat top.
Yeah, I don't know if that would develop as it gets older. These are very young. Clavaria dolphus is very large, growing up to 12 inches tall.
Yeah, we can see that they start out small.
But the way that it's broadening out at the top points me away from Pistolaris and Americanus. I don't remember if I have good photos of Ligula or not, but Ligula is another species that we've got here.
We could at least, this would be one where if I were you, Jessica, I would upload it as Clavaria delphis.
Yeah, keep it at genus level on this one, for sure.
And maybe take down in my notes, Clavaria delphis pistolaris. Spell it wrong a bunch of times and add a question mark. That is exactly what I would do.
Does someone want to answer our favorite question about this mushroom?
It is edible.
Can you eat it? Yeah.
Can you eat it? Yes, I guess. Is that what you said?
Yeah, they call them sweet clubs.
Sweet clubs.
I've heard the flavor. I've eaten them. They're okay. They're not that bad. Their flavor is described as wet rope.
Wet rope.
Rope. I don't know who the hell came up with that. I've eaten them. They're not bad. They're not spectacular, but yeah, they're edible. I would have them again.
So I would say that's
wet rope.
Wet
rope. I mean, I kind of like the smell of like wet hay, like fresh hay. Is it that kind of like sweet? I don't know if I would like the taste, but I'd like the. Well, when you can't eat it, can you make a tea with it? Probably. I don't know. Do you like it? Could find out. Yeah, so that's a cool mystery. Also, beautiful photo. I love the arrangement of just these three little mushrooms. And I'm not super familiar with it, so I'm going to go back, and one of my other very favorite things to do when getting into a mystery in an area that I don't know that much about is to look at the taxonomy. How are we sorting this, at least based on what iNaturalist calls the taxonomy? Okay, so Clavaria delphis, the genus, is in its own little family, Clavaria delphaceae. Let's pop up there. There's only one other genus. Haven't heard of that one either, but that says more about me. Wow, that's different looking. It's got teeth.
Yeah, I think this hails from like New Zealand or Australia, this genus.
Oh.
Pretty bizarre one.
Familiar with that one?
Yeah, you're right, Brennan. Somebody's been looking at this before.
This is what I do.
Yeah, me too. I just haven't run into this one. That's cool. I wonder if people look for hedgehogs, this would be one that if you're in New Zealand, if hedgehogs are also in New Zealand, something else I don't know about New Zealand, that would be definitely one I would be confusing unless there's a texture difference. Interesting. Also goes to show if I were to go to New Zealand on a trip and I saw that and I was just like, oh, it's a hedgehog mushroom, it's hidden. I might get it wrong. It's probably not going to hurt me, but when going to a totally different place, there can be totally different mushrooms. Order Gomphales is what they're all in. And then we go to look at what else is in that order. And this is where it starts to get cool as far as mysteries go, that you start to see just this big, what immediately feels like a big variation, but you can start to see kind of what's similar. I almost feel like there's a texture similarity here. That kind of, I don't know how to describe it, gummy, matte texture. And of course, forming either a coral or a club shape, but then there's these outliers that are more spiny. Beautiful mushrooms. Okay, so that's why it's really fun to go to the taxonomy tab and then keep looking. And then you find out Gomphales is in this group with stinkhorns in it. That's amazing too in this subclass. So you kind of can just follow it up and find out who's related to who. Okay. Were there more mysteries in the chat, or I could go back to the...
That's the only one I saw. If other folks want to kind of dig through their photos and throw something up for us to look at, go ahead and give it a try, but nothing else.
Yeah, I've got another one that I think will be of interest to a certain other mycologist at UW-La Crosse. So this is one that's up for sequencing for us next. It's in the ones that Tavis provided to us. He put it as Hygrophorus section Podorini, and that is, it's a waxy cap.
I think I actually put it in as Puterinus, and I was corrected by someone on a different species suggestion that blended the two and put it in section Puterini.
Okay, so you had gotten to species and then you were like, oh, maybe I can't.
I had gotten to species and somebody offered a different ID.
Okay, cool.
Not research grade. And I don't think it's his either. I gave the closest name that I could based on the old Smith and Hessler monograph.
Yeah, and you said it was growing with balsam fir, so I thought this was
balsam fir at Jack Lake in Langlade County.
And found in June, early June.
Early June. We were on a morel hunt, so it was pretty early fruited.
Yeah. I'm wondering if Lauren Ray has anything to say about this mushroom. I don't know if you can talk, Lauren.
It's got rice.
I don't know. Can you hear me from the
Yeah.
I don't know anything about Hygrophorus, so the end. Sadie,
It looks cool. I've never seen a Puterini, and I'd like to.
It was beautiful. There was quite a quantity of them too.
It's very gorgeous. We're admiring it from the big screen here at WWL. It just took us this long to get our computers to work properly, so we're just tapping into the taxonomy right now.
Yeah, so the rosy disc on the one on the far left was one of the identifying features that helped me lead to Puterinus.
Oh, okay.
And its association with balsam fir. I mean, to me, the description that I found was Puterinus, but I think it was, I can't remember his name. He's a European guy. He's writing one of the Mushrooms of Europe books. The species he sent, if I'm not mistaken, was a European species. And I don't know if he looked to see that I was in Wisconsin or not. I can't think of what his name is. But that's the suggestion he offered.
I can, if it was on the iNaturalist post, I can go to it.
It's on there for sure. Yeah, he's.
Oh, this guy. Yeah, he's on iNaturalist a lot. Nicholas Schwab.
Yeah, he's very active.
Yeah, so let's see. This you had put Hygrophorus Puterinus, and I did just realize that apparently a common name for these is woodwax. They are in the waxy cap family, but we could also call them woodwaxes. So you had put it in this. And I can see why. They're stout. But they're pink, but I guess you were like, well, maybe they vary in color. Was that the concept?
The rosy disc, I guess looking at these pictures, I wouldn't say that these pictures match the Smith and Hessler monograph.
I see. Yeah, sometimes there ends up being like a concept people build around things based on field guides or names. Like you say rosy and everybody thinks pink. And only looks at the pink ones. Although now that I'm looking deeper, there's definitely ones that are being put that looks like a...
That to me is what I found right there.
Yeah. So this might be, of course, that it's in this species concept, the Hygrophorus Puterinus.
The name that he offered is also in section Puterini, and that's why it's blended those two together. So we're both agreeing that it's in that section.
Yes.
And it could be completely undescribed. We'll find out.
Yeah. Yep, that's the other option. It's like it could be that there's just these variations and these are all this species, or it could be that some of these that are getting put in it, maybe the yellow ones, maybe if that's diagnostic, maybe it's totally different. He was putting it in this, and yeah, I see immediately what you're talking about, that I guess it's not really being seen in Europe, but it's
not
barely
European
being
green,
seen
no.
here too.
Oh yeah, it was actually I did some reading on that one. It was a rare sighting, and the ones that I looked at were from Quebec.
Yeah, so honestly,
And
that might
that
be
to me
something
is
else.
not the same mushroom.
Yeah, I can. There's also this granular texture on the top of.
Yeah, I'm not saying that I'm right. I disagree with his. That's why I left it alone. Section Puderini is good enough for me for now.
You got some folks in the waiting room.
Hi, Jeremy.
Hi.
Hey, Mariah, I don't think anyone can hear you. I think I accidentally muted you. That's the problem. I was trying to admit someone from the waiting room, and the button changed, and I muted you instead.
Okay, great. Sorry, I forgot I had to actually go there. Kept asking you to do it. Whatever.
Sorry.
Great. Back to the mysteries. I was saying this is a beautiful Rushula, but you guys didn't need me to say that. It just obviously is. This is also a great example of a good thing to do when you take a photo of a mushroom that you're trying to put up for ID or show to other people. Show it with its context. Like this one, Tavis doesn't even need to say what trees this was with. We can see the needles right there in the background. I can even count them and see that that is a white pine needle.
White pine associate.
White pine associate. I was curious, Tavis, that you didn't put a section on this one because you have taught. Like, is it that it's ambiguous, or you were just uploading faster?
With Rushula, subgenus Heterophyllidea and subgenus Rushula can be really difficult to distinguish, and I know it's in one of those two. And that's
Okay.
as far as I could get with this was subgenus. I'm sorry, genus. Subgenus.
Yeah.
I would expect it's in subgenus Rushula, but it
Okay.
could very well be in Heterophyllidea.
Okay. So that answers that. I kind of assume that it must represent a toss up because you know the
Definitely, yeah.
And yeah, if anybody had, I mean, this one is a hard one, and if Tavis doesn't know it, and he's a Rushula guy, but you know, we have a lot of people here who might be have put in your thought, put in your like, what would you have called this in the field?
What would I have called it?
Yeah, if you were forced to do a species on it.
So what's tough with Rushula too is there's some species that can be yellow or purple.
Wow.
So could it be Rushula peckii? It seems a little orange for that, but it could be. I don't think peckii is usually a lot redder than this. It's not fulvescens because it has a blush on the stem. It could be olivacea, which can be anywhere from olive green to brown to orange.
Wow.
Yeah, Rushula is terrible for identifying on morph only.
A morphological type, and that's also why this is an exciting one to get a sequence on.
Yeah, I'm very curious on this one, and I've got a huge Rushula collection here of literally hundreds of samples that I'm kind of reserving. I didn't want to send those in the first place.
Yeah, absolutely. That's the Rushula Reserve. This one was one that made the plate. Also, somebody cut me off if we hit 7 p.m. just so we got 5 minutes. But this was a really exciting one. So much so that Tavis has put it under the scope to try to get more on it. Both its sort of general look, correct me if I'm wrong, but I think the scalloped edges there seems to be kind of a Lulesia thing for some of them.
Usually, Lulesia has a cracked cap, with like Lulesia mundula. But
Mhm.
I know it's in the Entoloma family, but the spores were not angular, so that kind of pushes me away from Entoloma proper.
Okay.
And I think I have micro photos of this somewhere, maybe. But I believe
Cool.
that these spores were ellipsoid.
Ellipsoid, okay. And one of the things I was curious about is, does this right there, kind of on the farthest left individual, does that represent staining, like on the gills there?
So I've got another one. I know we're short on time today. I have another Entoloma that was found in the same woods that stained very easily blue.
I think I know which one you're talking about.
We just found it last year at Giant Pine.
Yeah. So I'm gonna.
You don't probably have to scroll back too far.
No, I'm also gonna narrow it down just by looking at Entoloma ACA. It's this one, right?
Right there, yeah, that's the one.
Once again, gorgeous mushroom.
Oh,
Yeah.
I remember that one. That was so weird.
It was bizarre, yeah.
And so you were thinking like the slight staining on that one and the staining on this one.
This one
Like
had angular spores.
Okay, so it's definitely
This
different.
one's Entoloma, yeah. I have that one collected. It's going on the next plate.
Yeah, but I'd seen this one too when I was looking through your Entoloma family
Yep.
observations, and that's partly why I was seeing this, and I was like, is that staining, or is that just color? There's also kind of a bluish cast right there to me in the photo, at least, and same for right.
And it could have just been no, because the spores were different. They'll both get
Right. It's not the same thing, but like,
Not
yeah,
the same
just
thing.
I was curious if it was staining. I'm gonna go to some next ones because I want to get to the disagreement about a deer mushroom, but also wanted to highlight basal mycelium. Just nice to see, and a term that people who are getting into mushroom ID might want to take a look at. When you dig out the thing, you can see the mycelium sometimes at the base, and sometimes there's characteristics about it, whether it's colored in a particular way, or it sometimes has a specific smell, sometimes is thicker or has micro features. So just something to look at. This one I'm gonna skip because I want to get to the deer mushrooms, but I'll just say Hebeloma. We're curious.
Probably Hebeloma mesophyum.
Okay, great.
Or
We do have
Hebeloma velutipes. They're hard to tell apart as well.
velutipes and Hesophyllum. You said
Mesophyum.
Hesophyum. Okay,
Yeah.
cool.
We've got more Hebeloma here than people think. There's quite a handful of species here.
I think many people underestimate Hebeloma in Wisconsin. Ever since I learned how to even have that as an idea, I see them a lot. So I agree with you there. This one's a huge mystery, I'm assuming, because you have it at Agaricales, which is a order. So it's pretty high up in taxonomic.
I think this is Typhrasia.
Okay, is that a genus?
Yeah, but
Not a genus, I know.
But I can't say for certain. This was a really bizarre mushroom. It was the only mushroom I found this day. It was near River Falls. Well, I found an asco. I found a Japhnia. But it was a very dry day. It was on wood. I believe it to be Typhrasia. Looking forward to results on this. That's all I can say.
Absolutely. So I'm going to skip this deer mushroom and go to the one that we disagreed on. All right. So what are we calling it? This is Tavis in one corner. Pluteus from
I called this one Flavofluogineus is what I have it in as my notes.
Oh, then you don't disagree at all.
No, I don't.
Okay.
This is certainly Flavofluogineus.
Okay, you just maybe accidentally to put it on right. Okay, no disagreement, no fight, and we're at 7 p.m. anyway. We're going to learn a lot from Alfredo about how to ID beautiful mushrooms like this. If you were calling this the same thing as this, now you're going to learn more and also probably learn a lot about the limits of our knowledge. My last thing was I was just going to draw attention to that this one has a rose comb, I think. Last I heard a viral thing or bacterial. Don't know. I'm going to move on. That kind of And so we are at 7:01. Going to introduce Alfredo Justo. And here's his bio. Alfredo is the curator of botany and mycology at the New Brunswick Museum in Canada. He oversees the curation and growth of the NBM herbarium, which comprises over 100,000 specimens of fungi, plants, lichens, bryophytes, and algae from New Brunswick and neighboring regions. His research focuses on the fungal diversity of New Brunswick and Atlantic Canada, while maintaining an ongoing global taxonomic research on selected genera of mushrooms. Alfredo completed his PhD in systematic mycology at the University of Vigo in 2006. Following several years of projects in Spain related to mycological conservation and diversity, he spent 6 years in a postdoctoral research position with Dr. Dave Hibbett at Clark University in Massachusetts, focusing on molecular systematics of mushroom-forming fungi. Research and teaching positions followed in Mexico, Spain, and eventually back to the USA, where Alfredo was a visiting assistant professor in the biology department at Worcester State University and a visiting scholar at Clark University. You can check out his publications at his very own website, or find him on iNaturalist as Alfredo Justo. All right, I'm going to stop sharing my screen, and Alfredo can start. And
Hello,
Pluteus.
everybody.
Here we have Lisa in the chat going plute baby, and that's cute.
I'm gonna first share this. Can you hear me okay?
Yeah.
Okay. So I'm gonna first share the screen and make sure that everything works. Here's my... Okay. So you can see the screen okay. You see the slides moving. Perfect. So thank you so much, Mariah, for the introduction. Thank you so much, the Wisconsin Mycological Society, for inviting me. I'm going to tell you a little bit today about the genus Pluteus in North America, and I hope I can get you more interested in these tiny creatures of the forest. Just a couple of things about myself. I'm now the curator of botany and mycology at the New Brunswick Museum in St. John, New Brunswick, in Canada. Our public-facing part of the museum is currently closed because we're under renovation, and the big buildings that you see there, this is the original building of the museum. There is currently being renovated, and this doesn't exist yet, but this is going to be the new museum scheduled to open in 2028. So if you're ever in New Brunswick, stop in St. John and check out the New Brunswick Museum. And what I do is one of my main responsibilities, other than taking care of the collection and curating the collection and all its different aspects, is study the biodiversity of mushrooms in New Brunswick and in Atlantic Canada and neighboring regions of the US. We are part of the MycoMap network, as everybody should be, and we have the MycoMap Atlantic Canada network that includes New Brunswick, but also our neighboring provinces of Nova Scotia, Prince Edward Island, and Newfoundland and Labrador. And before I start the presentation today, I just want to, and I will be reminding this again and again during the presentation, but I think it's very important that I'm presenting today, but the work that I do is only possible through collaboration. Collaboration with people working in other institutions, other researchers, but a lot of the times people like you guys that are part of mycological societies and are not necessarily professional mycologists, but without the work that you do and without the collections that you make, the work that I do would not be possible. So this is a list of people who actually contributed collections to the things that I'm going to be presenting today, but there are many, many more, and the number keeps increasing every day.
I think one of those people is here in our audience today, just so you know.
Yes, and are you talking about Lauren?
Oh, Owen Zeka, but maybe Lauren too. I don't know if she's
Lauren, I have one of her collections. Owen also has sent me collections, but I don't think I'm gonna be talking about his today. I was thinking about Lauren because one of her collections, one of her photos is in the presentation. But yeah, I'm sure there are probably even many people who might not be even in the list. But yeah, again, the work that I do is only possible through people who collect interesting stuff, and then I get to study it. So what is Pluteus? Well, Pluteus is a genus of mushrooms and it's relatively easy to recognize. It's saprotrophic, so what it does is decays organic matter. Most of the time you're gonna find it growing or fruiting directly connected to wood. It has pink spores, so the gills, when they're young, they're white because their spores are not mature yet. But as they grow, the gills turn pink because the spores are pink. And as you know, there are not that many things with pink spores. The only other big genus is Antoloma, but obviously you're going to find out if it's a Plutus or an Antoloma very quickly when you put them under the microscope. And it has been estimated classically that Pluteus has around 150 to 300 species. Currently, in our phylogenetic studies that by no means are complete at all, we already had uncovered 250 species of Pluteus worldwide. So probably we're looking at least at 400 to 500 species of Pluteus when you consider the whole diversity in the world. One thing that is important to know about Pluteus, and you're going to hear me saying these names through the presentation, is that Pluteus is divided into three major groups or sections. Section Pluteus, section Celluloderma, and section Hispidoderma. And the way these groups are characterized is how their cells in the cap look like, and how the cystidia, and the cystidia are just specialized cells that occur in the gills of the mushrooms, and how those cells look like. So in section Pluteus, the cells on the cap are cylindrical and they are completely flat, as you can see. And the cystidia on the gills are what we call metuloids, which are thick wall cystidia that usually have some ornamentation on the top. In section Celluloderma, the cells on the cap are rounded and they do have cystidia, but they are not metuloid. They are not thick wall. And then you have section Hispidoderma, which also has cylindrical cells, but instead of being flat, like in section Pluteus, they're sitting upright on the cap. And that's usually why the species in this section externally, they do have like a velvety appearance. And the cystidia, again, are not metuloid. So our DNA work on Plutus really started like going in 2011. So that was not that long ago, about 15 years since we published our like big first snapshot of DNA studies in Plutus. And at that time, we included 98 DNA sequences of the fungal barcode, the ITS. And at that point, in those very initial phylogenies, very preliminary phylogenies, we were able to recognize 25 species or species groups occurring in North America. But again, it was very, very preliminary. What we were looking mostly was, do these sections that I just introduced, Pluteus, Celluloderma, Hispidoderma, do these sections actually are supported by molecular data? And pretty much they are. I would say there are a few exceptions. There's a few oddballs that do not fit nicely anywhere. But I would say for 90% of the species, those sections actually are supported through the phylogeny. But there were many unresolved questions after these initial phylogenies that we produced. It was very difficult to kind of establish a correspondence between published names in Pluteus, historical names, and we're going to talk a lot about those in the presentation, and the actual clades and the actual groups that you see in the phylogenetic trees. So we decided to start going kind of group by group and looking more closely at the different groups, at the different molecular lineages that we could see in the phylogenies. And we started with what is probably the more commonly reported species of Pluteus around the world, And if there is a Pluteus species that is going to be in a field guide, it's probably going to be this one, Pluteus cervinus, or the deer mushroom. Cervinus is from the Latin cervus, that means deer, and that's where it gets its common name. And Pluteus cervinus has been reported all over the world, but there are also additional species that have been reported both in Europe and North America. So one of our first questions was, is really Pluteus cervinus a species that occurs all over the world? So we try to answer that question. And this is trickier to do in online presentations, but I try to make you think. Like, these are all 6 collections that were actually in our study of the Pluteus cervinus groups. And you can see some morphological variation here. You can see some that have kind of orange color, some gray color, a different shade of brown, some that are completely white. So there, you see some variations. And I want to make you think for a second, how many species do you think are represented in this set of collections? Is it all one species? Do we have 6 different species, or do we have something in between? Think for a second. When I do this presentation live, I always wait for the answers, but I'm just going to let you think for a second. And I'm going to give you another puzzle. In this case, other 6 photographs. Again, all 6 of these collections were included in our study of the Pluteus cervinus group. And you can see here that kind of this less variation when you go from photograph to photograph, and they look pretty much the same. So I'm going to let you think for a second. Again, 6 collections here. How many species do you think we have? 6 collections here. How many species do you think we have? And the answers is that the first photograph that I show you is actually all one species. It's all Pluteus cervinus. All that morphological variation that you're looking at in those photographs is morphological variation that happens within one species. On the other hand, the second photo that I show you that shows all these collections that look pretty similar to each other, they actually represent, each one of them represents a different species. So there are 6 species being represented there. So if you're just looking at morphology, it's going to be very hard for you to get to the answer to what is the taxonomy, what is the species diversity of this group. So we set out to study this group more in detail. First of all, again, this was a collaboration with researchers in Europe, in North America, both professional mycologists and amateur mycologists. It's all based on collections. We were trying to study and sequence as many collections as we could, including type and original collections. Types, and I'm going to be repeating this word a lot during the presentation. A type is a reference collection for a certain species that usually was designated when that species was originally described. And the taxonomy that we try to do tries to integrate all this information. We look at the external morphology, we put the thing under the microscope, look at the spores, look at the different structures that we find. We look at the ecology. In the case of Pluteus, for example, if something is growing directly on wood versus growing on the ground, there are a few species that actually don't grow connected to wood. Is it growing on a particular tree species? Is it growing on a hardwood or a conifer? And we're looking, of course, throughout a very vast geographic area. We were looking at collections from North America and all over Europe. And since we knew that ITS alone was not going to, the barcode, the fungal barcode, was not going to give us all the answers that we needed, we were looking at a different gene to use together with ITS. This is TEF1, which is a gene that is commonly used also in phylogenies as kind of a complement, a secondary barcode for Pluteus. And these were the results, and this is what we published in 2014. This was the kind of the taxonomic and phylogenetic study of this group all over the temperate areas of the Northern Hemisphere. We recognize 26 species in the phylogeny. This is kind of a very schematic representation of the phylogeny that we produce. 12 of those were described as new in the year 2014, which is very, very recently. And for North America, we actually have 19, or at the moment of the publication, 19 species were reported for North America, and 10 of them were described as new, which means that half of the diversity of the group in North America about 10 years ago did not have a name to be able to refer to the species. And that is going to be a common theme for Pluteus. And as you know, as you obviously know, because you're part of a mycological society, we still have a lot of work to do in North America to get our taxonomy in order. One thing that we did find out is that, yes, some species like Pluteus cervinus are actually widespread and a few others, but we also have endemic species. And they're endemic usually at the level of region. We have species that occur in Eastern North America. These species here highlighted, and we have species that only occur in Western North America. And for some of them, we have recently found, for example, Pluteus elafinus, which was described from Massachusetts. There was recently, a couple of years ago, one collection sequence from Alaska. So now we know that this species is not only in Eastern North America, it occurs in Alaska, it probably occurs all the way in between these two places. But these species that I highlight in this slide have only been found so far in North America. And one thing that I think is also very important to highlight, many of the species that we were able to describe, we were only able to describe them because there were community science projects already happening and regional diversity projects already happening. These species, Pluteus orestes that I described with Elsa Belinga from California, all the collections that we use to describe these species were collections made by community scientists that we were doing a catalog of fungi of Yosemite National Park in collaboration with Elsa and the people at UC Berkeley, but all of the collections were made by community scientists. There was no collection made by a professional mycologist of these species. And Pluteus orestes, the name Orestes means mountain hiker because this is a species that only occurs in the mountain ranges of Western North America. And if you go to iNaturalist, these are currently all the observations of Pluteus orestes, many of them from the Sierra Nevada range in California, but also other mountain ranges across Western North America, and is known as far north as Seattle and as far south as the mountains outside of LA. These 27 observations does, as far as we know about this organism on planet Earth, that's where it occurs. It's a regional endemic of Western North America. And again, all this new information that is coming, all that we're learning about these species is happening because of, again, community scientists making those catalogs and making those collections and sending them for DNA sequencing. And that's how we're learning more and more about the distribution and ecology of these species. So describing a species, giving it a name is just the beginning. Then you have to learn a lot about what the actual biology of the species is. And our paper in 2014, you think with such a big revision, then, well, all of the work is done. The work is never done. Just a few years later, Terry Clemens from the Arizona Mycological Society, she found and she sequenced through the University of Arizona some collections that she was finding in high elevation conifer forest in Arizona, and the DNA was not matching with anything from our 2014 paper. So eventually she sent me the collections and we described the species together as Pluteus arizonicus. And this is the DNA results, the DNA phylogenetic tree. You see Pluteus arizonicus here. These are all of Terry's collections, and these are some other species that we have already identified in our previous paper. And we just published this in 2024. So 10 years after that initial paper, we're still finding new things. And after we describe Pluteus arizonicus, it's actually one of the more commonly sequenced species for all the American Southwest. So you can see it's common in Arizona. Again, usually high elevation conifer forest is common across the Rocky Mountains, Colorado, New Mexico, and we even have some records from California and from Nevada. And the work continues even today. This is a work in progress. Even after you kind of think you have everything done, you have revised all the taxonomy of a certain group, new data keeps accumulating. And this is a phylogenetic tree that I just created a few months ago with some new sequences. And what I have highlighted in color are things that still need to be described. These are sequences initially identified as Pluteus posarianus, which is a European species. You can see here the European sequences. Here are the North American sequences. You can see this big clade here is Pluteus parilis, which is another species that we already mentioned in our initial paper, but it has not been formally described yet because we didn't have enough collections, but now we do, and it's actually quite common. It occurs all the way from California to BC, and this is a slightly rarer species that is closely related. So again, even for this group that has been already the subject of a big monograph, you're still gonna keep finding new things. It gets easier and easier because you keep accumulating more reference data, but the work continues even for these groups. So I told you a little bit about section Pluteus, and I told you about our paper and the follow-ups and how we're still doing work. But as I told you in the beginning, there are two other sections, two other big groups within Pluteus, section Celluloderma and section Hispidoderma. And these two sections is actually where the diversity in terms of a species actually is. Section Celluloderma, we have more than 150 species worldwide, and in section Hispidoderma, more than 60. And this is just looking at the phylogeny. It's the species that we recognize looking at the DNA data. And the phylogeny is, they're still missing whole regions of the world. Most of Africa is missing. There is a lot of tropical regions that are undersampled. South America is still very undersampled in comparison to North America. So we haven't even begun to cover in detail those regions. So this number is going to keep increasing. And you can see these color boxes represent lineages or clades or groups of species that for convenience, we have given them some informal names. And for the most part, these groups are not only phylogenetic groups or DNA groups. The species that you can find inside each of these color boxes are actually species that share some morphological characteristics. So how we decided to approach the study of these sections was the same way that we did our initial study of section Pluteus. First of all, collaboration, and you can see people here like René Leboeuf from Quebec, Stephen Russell, of course, who has contributed so many collections and sequences. It's still collection-based. The basic unit for our study is having as many collections as we can, both modern collections, but also original type collections. And again, we're trying to put all the pieces of the puzzle together. We're looking at the morphology externally, we're looking under the microscope, looking at the ecology, the geographic distribution, and we keep using two different genes, ITS, our fungal barcode, which is still pretty good and is gonna give you a lot of information, and TF1, because it's gonna give you the extra information that ITS is not gonna be able to give you. And this, I took this snapshot from my naturalist yesterday. If you look today, the numbers have already changed. There are already more sequences. These are all the sequence collections of Pluteus in North America. We're currently over 2,000 sequence collections, and you probably know about Mycota Labs and the MycoMap projects. That's where a lot of the sequences are coming from. Also from the Ohio Mushroom DNA Lab, Danny Miller, in the Pacific Northwest, also doing sequencing. So again, it's community science that is moving forward our study of fungal diversity in North America. And 2,000 collections is a lot of collections and it's a lot of information. But one key aspect that we have to think about when we study things in North America is the species that were actually described from North America, from the early North American mycologists of the late 19th century and early 20th century. William Murrell, Charles Peck, George Atkinson. These three in particular, they actually, especially Murrell, they actually describe quite a few species of Pluteus, and they describe it based on collections made in North America. But many of the names that Murrell and Peck and Atkinson, not all of them, a few of them have been used since they were described, but the great majority of names that they propose, they have laid in obscurity for more than 100 years. We have not been using those names for the North American collections of Pluteus. And even when we have been using them, there has been some taxonomic confusion about what exactly those names mean. So before I continue giving you some updates on new results, we're going to take a small detour down history lane of Pluteus in North America and why it's so difficult to see those names proposed by Murrell and Peck and Atkinson used by mycologists over the 20th century. Rolf Singer, a towering figure in 20th century mycology, he actually studied Pluteus a lot. And in 1956, he published his contributions towards a monograph of the genus Pluteus that even had a worldwide key for the genus in 1956. And what Singer did was for each species, he studied original and type material if it was available or his own collections. And then for each species, he will give some notes about the morphological characteristics. In this case, he's studying some species that Murrell described, Pluteus alachuanus. And these species, he measures the spores and then he gives some comments. And in this case, he says that he thinks that this species is actually a synonym of another species. So you can start seeing that, yes, we have the initial description by Murrell, but then another mycologist comes along and then starts having some taxonomic opinions about what the species actually is. Just a couple of years later, in 1958, Alexander Smith and Daniel Stans, they published their own revision of the Murrell types. And one of the funniest things is that, in theory, they're looking at the same collections that Singer was just looking a couple of years before them, but in many cases, they don't come to the same conclusion. So you have the original names, the study by Singer, then you have the study by Smith and Stans, and then what you keep increasing is the taxonomic uncertainty about how those original names, but Murrell and Peck and Atkinson should be applied. In the 70s, we have another important figure in the study of Pluteus in North America, Richard Homola, professor at the University of Maine, and he published a relatively modern monograph of section celluloderma in North America. And the way Homola interpreted these species and these names is going to set the tone and establish the concepts for how we keep using these names today. And a very important figure in the late 19th, early 2000s was Walter Sandberg and two of his PhD students, Partha Banerjee and Andrew Minnis, who studied Pluteus through several, again, revisions again of the original material, but also additional collections and new insights into the genus. And Andrew Minnis published his monograph of section celluloderma in 2010, which is 1 year before we started publishing molecular studies on Pluteus. And it's kind of a very nice transition because Drew Minnis then, he was a co-author with us in the first phylogeny. So from this long line of North American Pluteologists, Drew Cano is the link between those traditional taxonomists and the molecular phylogenies that we started doing in 2010. So we have those species, those names described by Muriel, by Peck, by Atkinson. We have those original collections hosted at the New York Botanical Garden, New York State Museum, University of Florida, Michigan University, Cornell, and the Field Museum. We have all these revisions, but those revisions, in many cases, more than giving us clarity about how these names should be applied, they're giving us confusion about how this name should be applied. And also very important is that for many of these names, for many of these species, other than the original collections, there are often very few, often zero collections made in between those 100 years since they were described. So it's very hard for us to connect the original names of Muriel, Peck, and Atkinson with the DNA studies that we're doing. But we're going to try anyway. So for section celluloderma and section hispidoderma, there are 3 groups for which we already have published papers with our best attempts are interpreting and putting those old names back into use when it's possible, attaching them to certain groups of DNA sequences that we see in the trees. And I'm not gonna tell you a lot because for these, the papers are out there, you can read them. This was our revision of the Pluteus leoninus group, where in this case, for example, we were able to not resurrect because it has been in use, but finally put the name Pluteus flavofuliginius as the correct name for the Eastern North American Pluteus leoninus species. Pluteus puma is the one that we describe for the Western North American species in this group. And in this case, we were actually able to sequence the type collection, the original Atkinson collection. We have a DNA sequence for Pluteus flavofuliginius. We also published already 2 studies in the Pluteus romeli group, which are the ones that usually have this bright yellow stem and brown caps, and the Pluteus podospilius group. And again, all of these studies are out there. You can look at all the details. I'm just going to give you some highlights about one of these studies, the study about the Pluteus podospilius group, because I think it presents examples of situations that keep happening again and again as you get into the taxonomic study of these organisms. So the name Pluteus podospilius is a species that was originally described from England. There is no type collection, there is no original material, so it's up to us, the people doing the taxonomy, deciding how this name is going to be applied. And in North America, there are 2 species that in this particular group that have been considered closely related to Pluteus podospilius. Some authors even have put these species in synonymy with Pluteus podospilius, and these species are Pluteus seticeps, which is an Atkinson species, and a variety of Pluteus seticeps created by Andrew Minnis in his monograph, which is Pluteus seticeps parietis hystidiosus. And this whole group is section celluloderma, again, with these rounded cells that you see. But one thing about the podospilius group is that they also have these long cells in between the smaller rounded cells, which is what gives them externally the velvety appearance of the cap that you're going to see in some of the photos. So again, in North America, we only had 2 species of this group, well, 1 species and 1 variety of this group reported. So when we did the phylogeny, we find out that there is far more diversity in this group than we were expecting, as is usually the case when you dig deeper into any of these groups. And this is the clade, this is the DNA group that we think is the best to keep the name Pluteus seticeps. It includes collections from the region where it was described. It matches the morphological concept that we have been using for the species. But that species, that variety that I just mentioned, Seticeps variety cystidiosus, the holotype is actually all the way here. So this is not a variety of Pluteus seticeps, but is actually a separate species. And even though only 2 species in this group were mentioned for North America, we actually recorded 10 species in North America in our revision that we published just a couple of years ago. Pluteus seticeps, again, the Atkinson species and again, here we kind of follow the morphological concept of homola for this species and try to attach it to a particular group in the phylogenies. This is Pluteus cystidiosus. But we also describe quite a few new species. This is Pluteus absconditus. Again, this velvety appearance that you see when you look at the cap, the granular aspect of the cap, how the surface really breaks into small patches or granules. This is because of the construction of the cells in the cap. Pluteus milsii and Pluteus inexpectatus, whereas the other species that we describe from North America. But even when you do these studies, there were actually 4 species that we couldn't describe, even though we had DNA data, because we didn't have enough information. These species over here from Ohio, we only have this one collection. So we know the DNA is different, but we didn't feel comfortable describing it just based on one collection. These other species here from Indiana, again, we also had one collection only, so we didn't describe it. These species here from Mississippi, also different based on DNA. We didn't describe it. For the Mississippi and the Indiana one, we now have more collections and we're gonna describe it soon. And we reported 10 species in North America, but as I showed you before, the work really never ends. And basically, as soon as we published this paper, there was another species that we found through sequencing, in this case in Western North America. This is the photo by Lauren that I was mentioning, and I have her collection here with me to study, to describe this species. This is the common Western North American species in the Pluteus podospilius group. And most of the sequences are from MyComApp projects, the California FundThis project, and again, community science efforts that are really documenting all this diversity. And this only has a provisional name that is the name that you will see on iNaturalist if you look under the provisional name field, but it hasn't been formally described yet. Soon, I hope, later this year. So I told you about section Pluteus. I told you about the groups in section Celluloderma and section Hispidoderma, which we have already studied and have published some papers about. And what I want to talk about in the final part of my talk is tell you a little bit about what we're doing right now. And I'm going to use the example first of this group of Pluteus, the Pluteus nanus group, which basically includes all the small Pluteus that have brown, gray, sometimes olive color caps. Many of them, they don't grow directly associated with wood. A lot of them just grow terrestrially among leaves, but not connected to wood. And this morphological group, actually, if you look at the phylogenies, you have a species here, but also here and here. So it's a very diverse group, as you can see already, just from the size of those color boxes. And we're doing the study of this group again in collaboration with colleagues in Europe. So in many cases, we have to figure out at the same time the European and the North American names to make sure that we're not, because in some cases, you're going to have the same species in both places. So for this group, this is probably our biggest data set. Our biggest project to date is endless. Every time I look on iNaturalist, there's probably something new on this group. There are 93 species, just this group, just the Pluteus nanus group in the Northern Hemisphere. That's 93 species, and we are not done finding new things through sequencing. Of those 93, 33 have names because we have sequenced the type collections, or we think they are the best candidates to keep some of the older names. But about 60 of the species will have to be described as new. So that's a lot of taxonomic homework, a lot of work that we have to do. By bringing it back to North America, we have all these sequences, and many of these sequences that we have on iNaturalist are on this big phylogeny that you see here, and that I'm not going to show you in detail, but you can see the size of the work that we have to do. Coming back to North America, we have all these sequences, but again, we have these old names that we don't know exactly what they correspond to. This is one example. This is Pluteus californicus, which was described from Pasadena. And since the original description, then Murrell redescribed it again in his catalog in 1917. The original collections are preserved at the New York Botanical Garden. And in the past, it has been almost impossible for us to get DNA sequences from these old collections. And this species, Pluteus californicus, is one of these species that have this greenish olive color in the cap. So we thought we had one clade in the phylogeny that probably was that, but we were never really sure, again, because we only had the original collections to compare it to. But enter Mycota Labs and enter nanopore sequencing. And last year, it was, I think, early summer, I decided, well, I have tried to sequence the Murrell material in the past and it has never worked. What if I give it a try with nanopore? What if with nanopore now we can go back to these historical collections and maybe get a DNA sequence, and we don't have to guess anymore. We don't have to think that, interpret and try to overthink exactly what those old names mean. So I sent a trial run of 20 collections, both from Murrell at New York Botanical Garden and Peck at the New York State Museum. And after a couple of tries, we got DNA sequences for all of them. And that's a big, big, big game changer for us, being able to go back to these original historic collections and get a DNA sequence. And for Pluteus californicus, we were able to sequence the type collection and one additional original collection there at the New York Botanical Garden. And it does match the DNA sequences that we thought were the best candidate for that name. And these are some of the modern collections, all from California. You can see those green colors in the cap that were mentioned in the original description, and that's why we thought these collections are a good candidate for keeping the name. But now we know for sure. Now we have a DNA sequence from the type collection that matches the DNA sequences of the modern collections that we study. And again, so that was the Alfred McClatchy, the original descriptor of Pluteus californicus, Murrell, who described it again. And then these are the known collections, mostly Southern California, but we do have one just south in San Francisco. So older names are coming back to life. And they're coming back to life because now we have the ability to actually go back to these collections. And this is important because when we have the type collections, when we have those reference collections, we can move on in describing new species. This is a paper that was published just about a month ago. We're describing one species in the Nanus group, Pluteus ochroviridans, which is the species highlighted here. But the reason why we were confident in describing this species as new is that now we have all these type sequences for these older names, so we know we're not describing something that already has a name. So having this reference data in place is fundamental to keep moving with the taxonomy of this group. So that's what we're doing right now. It's happening right now. After that initial trial, I asked for the original collections of Muriel, of Atkinson, of Peck, and in total 149 collections of Pluteus, but also Bulbariella and Bulbo Pluteus, originally described from North America, and they have all been sent to Mycota. And for many of them, we already have DNA sequences. Some of them, they're still working on that. But that's what's happening right now. We're finally kind of closing the circle. We're going back to the original material, but we're not doing just a morphological study anymore. Now we have the technology. Now we have the ability to get a DNA sequence from those original collections. And this in itself could be a whole separate presentation, which I'm not gonna subject you to right now. But I do want to give you a couple of examples of some of the more recent things that we have been able to sequence from these North American types. And I think it's very important to kind of put in value all the work that these early mycologists in the 20th century that didn't have access to PCR machines or DNA sequencing, all the work that they did was good work, and being able to put it in value again is very important. So to close out the talk today, I'm just going to give you a couple of recent results about the North American types. This is one of them. This is Pluteus ludovicianus, which is a species that Muriel described from Louisiana in 1917. And this is the original collection, the type collection that is in the New York Botanical Garden. This is how it looks like. From 1917 until 2010, that was still the only collection known of this species. This species, Drew Minis and Walter Sandberg, they did include it in their monograph of section celluloderma. It's in the dichotomous key. And when you go under material examine, there's only one collection. And they say it very clearly, this species is only known from the type collection. But they still thought that it was worth including because they thought that this is probably a good species. This is probably something different from the other species of Pluteus. But again, over 100 years since the original description, and still only one collection known. But we got a DNA sequence for the type. And we know now that that DNA sequence matches many modern collections that we do have through Mycota, through the Mycota project, and some of the sequence that I have done even before Mycota. This is a collection from Steve Russell. This is a collection actually from Costa Rica, from one of my collaborators in Mesoamerica, Osbert Morales. And these are all the first photographs that we have of these species over 100 years after it was originally described. So this is another resurrection of a name that has been forgotten for over 100 years. And these are if you go to iNaturalist now, you can search for these species and you can see the photos of the collections that people are making. And these are just the sequence collections. So these we know for sure correspond to the actual species that Muriel described. And before we knew it was Pluteus ludovicianus, we were using some of the provisional names that we are using for DNA sequence collections. And the last example is to show you that we didn't always get things completely right. We did our best, but we didn't always get the things completely right. This is Pluteus harrisii, which is another Muriel species that he described from Jamaica. And this phylogeny that you see here this was published in one of our initial phylogenies in 2011. And we had some of the collaborators from Brazil. They had some collections that they corresponded morphologically pretty well to what Muriel had described. These are all the drawings based on the Brazilian collection. So we called it Pluteus harrisii because we thought that's probably what Muriel described, seems to match very well our own type observations, the original description. But now we have an actual sequence from the type, and these are the Brazilian collections, and this is actually Pluteus harrisii. So we were not too far off, but probably the Brazilian collections are not Pluteus harrisii. What we do have is many collections on iNaturalist, mostly from the Caribbean, that do match the sequence that we now have for the holotype collection, the original Murray collection of Pluteus harrisii. And now we know how this species looks. We actually have color photographs that we can look at and get a sense of how these species actually look like. Again, it took us 100 years, but we finally got there. And these are the confirmed occurrences of Pluteus harrisii. We have quite a few of them from Florida. We have some from Puerto Rico, but it seems that it's mostly a subtropical species here in North America. And I wanted to, if you want to have a really, really, really nice look at the species, this is an observation of Pluteus harrisii by John Cook, the president of the Florida Mycological Society. He has all the macro photos. So if you go to the iNaturalist observation, he also has micro photos of the particular cystidia that this species has. So a really wonderful observation. Again, for a species and for a name that for over 100 years, we didn't know what it was. Well, now we do, and now we're able to put finally all the pieces of the puzzle together and have an updated taxonomy and bring all those older names back to life. So a big thank you again to all the people who have contributed collections, to all the labs that do the sequencing, Mycota, the Ohio Mushroom DNA Lab, Danny Miller, anybody who has contributed sequences. There are other people who also do sequencing on their own, anybody who has sent me collections. Every single bit helps of getting us there and having a more clear picture of the taxonomy. A big, big, big thank you to the institutions that hold these herbaria, who have these collections in an era with herbaria seen as something of the past that doesn't hold any value. Without the collections held and preserved and curated at these institutions, all the curators, the collections managers that work in these herbaria, without those collections, none of this would be possible. Would still be wondering what these species actually correspond to. So a big, big recognition to all the institutions that have these herbaria and keep them. And just to let you know that the photo that I've been using as my cover photo and my closing photo is a photo from René Leboeuf, and it's actually another one of these names by Muriel that we're going to bring back to life. This is Pluteus abellaneus, which is a species that Muriel described from the Adirondacks in New York, and we're finally, again, 100 years later, we're going to bring back to life and give it a full description. And that was all that I wanted to tell you about today.
Wow. Thank you so much, Alfredo. Like, that's probably the most like clappy hands I've seen in a like a quick succession, and so in depth, and also just hearing you talk or putting the words to it when you go back to the type specimens and you do the work to either sequence the type or to get something to come, like it's a resurrection, and that's really
Yeah.
really powerful to say it that way.
It feels really special to be able, and one thing that I really realized is how good of a mycologist Muriel was. And his descriptions are frustrating because they're very short and they're very vague, and they don't highlight the characters that I need him to highlight. But he had an eye for these things. He knew that something was different about them. He didn't have a DNA machine. He didn't have even the microscopes that we have today. But he knew something was different. And he didn't get everything right, but all things considered, all these North American mycologists, they did a really, really good job at taxonomy, and it's good to bring that full circle and back to life.
Or I've actually seen your talk before.
Yeah.
I had to watch it again. It was great. I have it
Yeah.
saved to my YouTube.
I changed a few things on this one. I think it
A
has
little bit, yeah.
more information on the types and some of the older names. So it's never exactly 100% the same.
Sure. Yeah. No, I saw some changes. Speaking on those descriptions, Merle was great. He was a hero.
Yeah.
He didn't do descriptions the way like Coker did.
No.
Coker's descriptions are fabulous.
Yeah.
They're the best. If anybody reads any Coker, it's awesome.
Yeah.
It'll paint a picture in your head of what you're looking at.
Yeah.
Davis, could you drop the name of the taxonomists you mentioned in the chat so that people can look them up because you recommended them?
Absolutely.
Thank you so much. I see Gosia has her hand up for a question.
I have some crazy mushrooms that grow on my property. So I saw and I took I have some dried samples here from the spring, and I just really would love if I could find some people to help me study these things. I've been posting them on the thing, and I'm so inspired by what I just listened to. Yeah, so I would love it. Yeah, come to my land and look at the mycelium. Yeah, I feel that the mycelium has been speaking to me, and I want to keep the mycelium going because that keeps us going, right?
Yeah. When you
Yeah.
said you're posting them, you mean iNaturalist or?
Yeah, I am. I'm a Daisy. I'm Daisy. Cheap Daisy.
Chief Daisy on there.
Yeah.
Cool.
Yeah, so I've been posting, but what he mentioned, I've been seeing these, but now I'm in Wapaka, which is near in Wisconsin. We're supposed to have snow tonight, but we just had a melt. So I've been studying like the moisture. It's the moisture, like the moisture, the temperature. So I'm like, all right, I'm waiting. I'm like, okay, all right, what kind of mushrooms might pop out? So, but we're gonna have a snow now. So I'm on pause because we had a nice warm up. So I've been following the heat, the moisture on my property, and the mushrooms that have been popping up when it's really warm. It's heaven. It's just heaven. I dance.
Maybe a question that could go to Alfredo would be, what are there deer mushrooms that are known winter fruiters or spring thaw?
Not that I know of. I think even the ones like Pluteus orestes that I mentioned we described from Yosemite and the Western mountain ranges, I don't think they're snowmelt mushrooms. I think they appear later in the season, more towards the peak season for the mountain, but there's no melt mushrooms. I know for sure that actual like fleshy mushrooms that occur, but I don't think any Pluteus does, at least for now.
Yeah.
Maybe one day we'll find one.
I think
Well,
yeah. Check out on your
I'll just keep posting on iNaturalist.
Yes.
I did the other day I saw some stuff and I'm like, let me just take a picture just in case. So I have a lot of artist conchs, a lot of those.
Yes, we are very rich in them.
Yeah.
I'll follow up about maybe some of your thoughts there.
That'd be wonderful. Yeah. Thank you. All right.
Thank you. And I see
mycelium.
mycelium, very true.
Yeah.
I see Ben King has his hand up too.
Yes, I was curious about when you're able to identify new species via DNA sequencing, is there a process then to go back and find ways so that those species can be described without sequencing further, like by morphology, ecology, and what is the process for that?
Yeah, at least in my case, the process is DNA is part of the puzzle, but it's not is what guides you. For me, is what guides you, but it is not what tells you this is a species or not. DNA is going to tell you, well, you have a group of organisms here with these DNA sequences, and this group of organisms here with these DNA sequences that are slightly different. At the end of the day, it's up to you, the person doing the taxonomy, to decide if these should be recognized as species or varieties or subspecies. The DNA is never going to tell you this is a species. It's just going to tell you, well, these things are different. It's up to you to look at the differences, to use phylogenetic methods to analyze them, to look at the morphology, to what I was showing you those slides where I was saying we try to integrate all the information. Everything is important to make that decision in the end to say, well, we have these things that have slightly different DNA and they have different ecology and they may have some differences in morphology, so we're going to call them different species. For me, it's never only the DNA. The DNA allows you to peek kind of under the hood and see what's happening there, how these organisms maybe, if they have differences in DNA, it means that they're no longer mating with each other. Probably they're different, the different paths of evolution. So we should be calling them different species, but it's never an absolute. At the end of the day, taxonomy is made up by us humans to try to understand the natural world and communicate about it. So I always try to, yes, find and highlight morphological differences. I always try in our papers that we have a dichotomous key that people can use. It's very difficult sometimes. The differences in morphology are sometimes very subtle. Sometimes we don't have enough collections to know how well those differences are gonna hold up as we collect more and more. It gets tricky, but I always do try to put morphology and ecology and geography also in the picture. They're as important as the DNA to make those taxonomic decisions.
Thank you.
Yeah, that was incredibly nuanced and very good. I have sort of an annoying follow-up question, which is that,
Yeah.
or maybe useful, I don't know.
Let's
So
piggyback.
yeah, piggybacked on this one that
Yeah.
that is incredibly nuanced and it is how it is. But we also learn, when you're actually like, okay, we're going to describe a species, we're always told
Mhm.
things like, well, in this group, 97%.
Yeah.
How do you feel about those sorts of lines, or do you develop those, or do you never use those?
It's never enough. I mean, I'm very wary of giving out those numbers. I think they're useful guidelines, but they're not absolutes. It's not that I can tell you in Pluteus, for example, Pluteus cervinus, Pluteus petasatus, other Pluteus species that are widely distributed on the northern hemisphere, Pluteus leucoborealis, their ITS can be up to 98, 97% similar, and you're still looking at the same species. And in those cases that I mentioned, I'm pretty confident that I'm looking at the same species because the morphology is the same, the ecology is the same, and we're using a secondary barcode, TF1, which is identical across all the collections, which is telling me that the differences in ITS are probably, well, coming from some, the more recent history. So I'm confident I'm looking at the same species. So up to 97, 98, 98% most of the time, I'm still looking at that. On the other hand, in the Pluteus nanus group that I just mentioned at the end of the talk, we can have a species up to 99% identical ITS. And then you look at depth and it's different. Then you look at morphology and it's different. Then you look at the geographic distribution and it's different. So, but you're still looking at 99% identical ITS. So I would call them different species because there are more things that are different than things that are similar. So it's never an absolute value. And at the end of the day, it's important to remember that DNA phylogenies recognize DNA lineages. What you call those lineages, whether it's species, varieties, subspecies, that's up to you. That's up to the person doing the taxonomy. And I think the only thing you can ask of a taxonomist is that they explain why they make the decisions that they make. And you can agree with them, you can disagree with them, that's completely fine. But you should explain, well, I'm coming to this conclusion because of these reasons. And then people who come later and maybe have more data, they can build up on that and change things and if they need to be changed. And just, I'm speaking as a taxonomist, but I feel that sometimes people don't realize how dynamic taxonomy is. We're trying to reconstruct millions of years of evolutionary history at the same time that we're trying to figure out names that were described 100 years ago. At the same time, we're trying to put a name in the collection that we made yesterday. And we have limited resources and limited information to do that. So we're doing our best. So I think the only thing you can do as a taxonomist is put all the information out there and explain why you made the decisions that you made. And
Yeah, I always challenge people to think about it like imagine you were just gifted a 1 billion book collection, but you only have access to the first 100 and you made your categories. Do you think they're going to hold up, or you're going to want to break up different categories? There's Todd Osmondson of UWS put a quote, a relevant quote: "A species is whatever a qualified taxonomist says it is." Yeah,
it's true. It's true. At the end of the day, like, if one of the Cortinarius people tells me this is a species, I'm not going to argue with them. I don't have the time to go argue with you. If they have made the work, if they have put in the effort to study those collections, and unless it's something completely, crazy, but most of the time, if somebody presents to me a taxonomic study that is well done and well justified, I'm not going to argue with the decisions that they made, because at the end of the day, what we want is put a name to the organism so we can learn more about that organism.
Yeah.
How widely distributed it is. Should we be concerned about conservation? Like the example that I put of Pluteus orestes is like, well, it's a relatively widespread species, but it only occurs in high elevation conifer forests in the mountain ranges of 3 states in North America. So when you think of it globally, it's not that common. If something happens, let's say forest fire, how is the species going to do? Is it going to do okay if there is more and more forest fires? Because that's where it grows on planet Earth. It doesn't grow anywhere else. So to me, that's why taxonomy is important. Now we have a name. So now we can follow up the species. Now we have reference morphology data, reference DNA data. We can learn more about the species. But yeah, at the end of the day, what I do is just the initial part, which is the taxonomy. But we need.
But it seems to involve ecology for you, though. Like you need.
It does to a certain extent, but I'm not an ecologist. I'm not doing an ecological study. I'm doing the very basic thing of telling you, well, this thing grows associated with conifer wood and it grows in this type of forest. And so far, we only have recorded that in a certain area with certain characteristics. So it's very, it's a starting point, but I'm not doing like population dynamics or ecology of the study. But again, we put these things forward, we give them a name, and somebody else can come after us and keep studying other aspects of the biology of these organisms.
Yeah. Speaking of people in WMS wanting to study these, we got a question from Max. A chat about it. If Max, did you want to unmute and ask, or I'm going to give it a minute and I'll say it. Okay.
I'm good.
Okay, cool.
We always
Okay, good.
put it at
In that case, he said, is there any variation among the spore color beyond pink or salmon if we're using spore prints to identify, like within Pluteus? I think that's a really good question, like because that's something that
No,
I think we do a lot is we can get a spore print in the field.
no, they're always pink, mycological pink, not bright pink, but it's pink. Is definitely pink. It's not brown like a Cortinarius or is yeah like those that salmon pink. The intensity of the color, like if you do a sport print, for example, for Pluteus cervinus or one of the small cellulodermas, the intensity is not going to be the same, but just because for one of the big Pluteus, you're going to get so many spores. One curious thing about Pluteus, once you collect it, even if the gills are completely white in the field, they're going to be pink by the time you're back in the lab. Pluteus start sporulating like crazy the moment they get severed from their substrate, immediately. And they decay very quickly from the moment you collect them. So don't leave them in the box forever because that Pluteus is going to be gone, especially the small ones. So yes, try to bring them back to the lab and dry them as soon as possible. Once they realize they're not in their happy habitat in the forest, they're like, I'm out of here. Start sporulating and the mushroom decays very, very quickly. So, but in terms of color, no, the color is, as far as I know, is always pink. The gills are not always pink. There are some species that have pigmented gills. There's some species with yellow gills. Some of the species in the Romelii group, they do have pink gills, but that's pigment of the gill itself. The spores are still pink. So there are a couple of things with color on the gills, but the spores themselves in Plutus, as far as I know, and as far as I can remember, they're always pink. Yeah.
Yeah, so it sounds like spore color is maybe not our best thing, but like you were saying, the more you pay attention to the characteristics, the more you have a chance of seeing. And in that vein, I've always been told, I think Tavis was maybe the first one who told me, Pluteus, you're looking at so free attachment that it's like a little racetrack around there. So extremely free gills. But is there ever any variation in gill attachment or lack thereof?
Not really. Some of the small cellulodermas, when they're really close, it may look like the gills are touching the stem, but not really. When you see them fully open, you can see the space between the end of the gill and the stem. Some of the small ones, when they're still not fully open, they might give the impression that they are attached, but they're really not. When you see the mushroom fully grown, they're always free. And yeah, and the free gills and the general aspect, I think, even just looking at the general aspect, the only thing that you would confuse them is with entoloma, but I think most entolomas just have a different look in the field. And even if you make the mistake, the moment you put them under the microscope, you're gonna realize really quickly what it is. So I think as genera go of mushrooms, Pluteus is actually to know it's a Pluteus is pretty straightforward. There are not that many things that they can get confused with. Now getting to species, that's probably you saw today is another different story. But again, it's not impossible. It's just it takes a minute, but it's not impossible at all.
I have some questions. Fredo, thank you so much for the fantastic talk.
This is Lauren.
Oh yeah.
Hi, Lauren.
Hi. My blood pressure just rose too when you were talking about collecting Pluteus because I've had multiple collections where I collected them and I waited too long to put them in the dehydrator, and then you've asked me for the specimen, and then I go to look and I realize I put it into dehydrator after it already started decaying and it's not good enough. So I do really, if you're interested in Pluteus, I really do recommend, because I'm someone who will put the tackle box. If I have a lot of backlog of collections, I have to put in the dehydrator or work up, but do not wait because they will rot. But thank you for just all the work you do on Pluteus. You're just a powerhouse, and all of us who love Pluteus are just so glad that you're doing the work you're doing. And what was I going to ask? Oh, and just thank you for shouting out all the community scientists who contribute, because they're all so proud and they're such a big part of these initiatives, and it
Yeah.
just motivates a lot of people to get out there and make collections.
Yeah.
But my question ultimately was, of all the different resurrections and resolutions you've had, was there one in particular that you were like most relieved to get an answer for, of all the ones you showed us or beyond?
All of them are important. Like some of the names that we didn't really know what they were are good because we can finally know. Like Pluteus ludovicianus, without a sequence from the type, is a small brown Pluteus. Like it could be anything. As I showed you, that group has over 90 species. There is no way that just by the morphology we could know what it is. So I would say I didn't go in detail today, but we also got quite a few things wrong. And there are going to be some adjustments in some of the names that we put forward before we have sequence for the types. All the sequence that were, every single sequence from types are exciting. Particularly relief. I don't know. Some of the things that I got right, like Plutus californicus, that I knew it was californicus before we, it's not relief, but it's like, hey, I can actually identify a mushroom. Look at that. It's cool to come to a conclusion and that have that conclusion confirmed by
Yeah.
sequencing the type. Plutus fulviparius, which we resurrected kind of in the Romeliae paper, we did get a sequence from the type, and it is what we call fulviparius, so it's great. But at the same time, we also have some things wrong. So yeah, what we call Plutus brunei discus is not, and now we have to change the name, and we tried.
Part of the process.
Uh, but no, all of the sequence. I'm trying to think if there's anything that. No, as I said before, what really overall struck me was how good of a mycologist Muriel was. And I've joked with some people that I'm going to have a T-shirt that says Muriel was right, and that's it. And that's like he got some things wrong, like everybody does, but overall, his eye for recognizing that something was different, considering the thousands of collections that must have gone through his hands through his life, is just amazing. The eye that he had to recognize, no, this is different, and in groups that are extremely difficult to separate even today. So that's my overall impression would be that Muriel was right.
Cool. It's a good summary.
Yeah. Any other questions before?
Right, you should really include in our web call for collections.
Sorry, you were cut there.
Oh, sorry. That was for Mariah. We should really have protocol for collections on our website accessible to everyone.
Yeah, well, we can work on that for sure. We definitely link to some good resources, like Patrick Laycock has the Myco Guide resources about it, but we should write up something
I
specific to
can write something. I'll do that.
Yeah. Thank you, Chief Mycologist Davis Lynch. Right on. Yeah, I was, because we definitely want to help contribute. That's a very practical question. I guess I have you highlighted how people can contribute through MycoMap and your project. That is something that our club could potentially connect with, I'm guessing.
Mm-hmm. Yeah, absolutely. Like just putting things on iNaturalist, well-documented collections, that always helps. I mean, the things that most of the time people find are relatively common things like Plutus cervinus, Plutus ongoi, Plutus petasatus. But once you get through the really common ones, there's going to be interesting stuff. Like we are not done finding all the good stuff yet.
I also a lot of times when we go out into the field to make collections, one of the harder things we have, I know I've failed to do it a number of times when I look back and I'm like, why? Is getting that, when you're the person on the ground, getting that ecological key data, like what, and is there anything that you would shout out for Pluteus that is like burning questions? This is what we want to know.
Conifer versus hardwoods, like if you don't have that information for like section Pluteus is fundamental. It's like, was it growing on conifer wood or hardwoods? You don't, most of the time you don't have to get to even know what particular species of tree you have, but just if it was a conifer or hardwood. And remember that Pluteus sometimes does not grow associated with wood, especially the small cellulodermas. A lot of the times they're growing directly on the ground. So know that and be aware of that, because if you think, oh, it's on the ground, so it's not a Pluteus. No, some of them, they usually grow like on, mulch or leaves or something, but sometimes it's not actual, a log or anything like that.
Oh, so it's not always buried wood. It's sometimes.
Yeah, sometimes they're actually, grow attached to the ground. So remember that if you're looking for Pluteus. But I would say in general, substrate, conifer versus hardwood versus terrestrial, and good photographs and put them on iNaturalist.
Amazing. Gosia, you have your hand up again. Hello. Oh, you're still muted. There you go.
Pine cones. I noticed like inside the little, like a little bit of nook got in there, a little numpch, and they started growing out of there. That's what I noticed. Is that true?
That's probably not Pluteus. There are a few other things, biosporas and things like that, that do grow on pine cones.
Diaspora and Strobilurius.
Strobilurius also,
Okay.
yeah.
All right.
So that's a good observation, because that's for some mushrooms that is going to be a very specific habitat that they grow on pine cones. So, noting that type of thing is always gonna be useful to identify the mushrooms. Yeah.
And I also noticed mushrooms growing inside of a dead stump, like in the middle, something like that.
Yeah, there I don't think is, well, I shouldn't say I don't think is that. Is more, I think, the stage of decay of the wood that I think is important, more than the fact that it's necessarily inside. Like Pluteus, for example, you're gonna find a lot of Pluteus, sometimes 2 or 3 species fruiting right next to each other. If you have big logs that have been decaying for a while in the forest, especially hardwood logs, that's a prime habitat for Pluteus. So really, really late stage decomposed wood is prime habitat for many species of Pluteus. So look for those areas where, if you have any, old forests with older trees that have fallen to the ground, they have been decaying for a while, that's gonna be a good habitat for Pluteus.
Are there other mushrooms that grow relationed to that mushroom? Like if I happen to notice like a wide variety of another mushroom, like they're cousins or like they're growing while, and this one kind of encourages it. It's just a theory.
I think there are mushrooms that share a preference for a certain habitat. So it's not as much as that they're kind of interacting with each other necessarily, but certain mushrooms, you tend to find them in the same type of habitat because they all prefer that particular ecology. So, yeah.
Surprise, surprise, I have another question, but I do want to make sure that other people get a chance to go who might not feel as empowered to just interrupt and go. So just like one moment. Did you have anybody else want to unmute, start, or ask a question? Okay, I get to ask mine. All right. It's kind of a follow-up on something that Gosia was saying about, and in your response, because when you were saying it, I realized absolutely, I've almost always seen Pluteus on those well-rotted logs. A lot of mushrooms have that almost successional preference for when they're going to make their moment, not only to maybe grow or be in that log, but also when is their moment to, like, I would assume, get a lot of nutrients and then fruit, like they might have been there, but then they're going now. Yeah. But it came to mind, the exception that I've seen kind of regularly is that the one couple of years we were seeing Pluteus orontorugosus. Almost every time it was like on a knot on a tree that was not that well decayed, that was maybe even alive in part of a tree, but decayed in one part of it.
Mhm.
And it was just, that's one of the things besides the fire trap cone orange color that almost had us being like, it's not Pluteus. And it was only looking microscopically and also seeing the free gills that we were like, okay, it is Pluteus. My first question is just kind of like, is that an association exception you've seen? And my second kind of ecosystem question is, I've been telling people, oh, I saw that mushroom specifically when there was a drought year and the year after that. Is that something that's in the air for Pluteus researchers or about Pluteus orontorugosus?
Well, for Arantirugosus, which probably we should start calling Caloceps in North America, but that's another story. Pluteus Arantirugosus, yeah, there are Pluteus that grow on wood that you stand in trees, wood that has not been through much decay. Pluteus petasatus is basically a ruderal species. It grows on wood chips, on logs, that stumps that have just been cut. So is that it's not that they only grow on well decayed wood. Is
Well, the
that that's
exceptions that prove the rule. Okay.
yeah, it's not much. I wouldn't say even exceptions. That there is a gradient of ecology. Like it's from the kind of completely like again, ruderal man-made habitats to really specific, super well-decayed wood, and it's only gonna grow on that. And there is all the gradient in between. And there is some flexibility for some species more than for others. There is some ecological kind of plasticity there. And yeah, was there another component to your question? Or
Yeah, it was just about the other ecological note I had for that species was that it really came out in a drought year and
the
the year after
drought, yeah.
drought year. Yeah.
I don't have a final answer on that. I can tell for mushrooms, there are certainly particular years where they fruit. For Pluteus, I will say last year was in New Brunswick was I think the driest summer we had since 1950 something, I think. And one of the very sad field trips that I did over the summer to forest that was crunchy, and there were Pluteus fruiting, and it was the base, and it wasn't like I was biased to find Pluteus. I was collecting everything. There were Pluteus fruiting, so I think, but that I think that ties back to the ecology because if they're going to be fruiting on really well-rotted logs, those well-rotted logs are going to be one of the few places where there is some humidity left to fruit. So I think it's not necessarily that they prefer the drought years, or maybe it's one of the few things that you're going to see on a drought year because their preferred substrate is going to be one of the few suitable ones to have to be able to fruit. So I think it's probably more in that vein that having a preference or
Yeah.
something for a drought year.
I think that's a really good point. I mean, the one thing I want to be like is, oh, well, my intuition is you're always looking for your niche. And if you're the only one, because of your happenstance of substrate preference, if you're the only one that can go off them, maybe you develop that more over time.
Yeah.
So,
Yeah.
but I'm sorry, there's a question in the chat related to my question. It's Randy Strobel asking, are Pluteus white or brown rotters?
They're white rotters. They're tricky little creatures because they don't grow in culture easily. They're very hard to grow from spores, which for saprotrophs is rare. I think there has to be something with some, internal dormancy clock with the spores because they're extremely hard to grow in culture. But there is a genome for Pluteus, well, in microcosm it says Pluteus serpinus, but it's not, it's Pluteus posarianus actually. But we do have a genome and they do have the enzymes, typical wood decay enzymes of the white rotters. So they're white rotters. They do, again, have a preference for more well-decayed wood than other white rotters like Trametes or something like that. But Again, some other species grow terrestrially. Some species like disturb habitats. So there is some plasticity through the genus, but white rotors, yeah. Oh yeah, white rotors there, yeah.
Just
They
put in a little
decay.
context.
Yes, yeah. So white rotors decay both the lignin and the cellulose, and brown rotors, they don't bother with the lignin, they go for the good stuff.
Yeah, and I feel like that's something that you can explore in the woods yourself if it's not always a one-to-one, but if you find a mushroom fruiting on some wood and you're pulling into that rot and you're finding that it's more like stringy or falls apart and it's whiter in color, it's paler, you might be like, white rot maybe. And then it's the enzymes in the genome that really tells some of the fuller story. But
yeah.
There's Max was also asking, are there any toxic species of Pluteus?
Well, there are psilocybin containing species of Pluteus, which is some kind of toxicity, I guess, but not in the sense of no, as far as I know, there is no Pluteus species that has amatoxins and there I didn't mention this, but Pluteus is relatively closely related to Amanita. They're in the same corner of the Agaricales. It's not the same family. They're quite closely related together, but there is no known species of Pluteus that I know of that has similar toxins to like Amanita or some of the toxins that are found on other agarics. The only kind of biochemical highlight is the species in the Salicinus group. They do have psilocybin. It hasn't been demonstrated for every single species in the clade, but it's actually one particular clade within section Pluteus where you can see the species that have psilocybin, but not toxicity in the sense of, yeah, toxic species. No, not as far as I know.
That's really cool. Plus, a lot of the context is interesting to know. I don't think I've ever thought about how close we're related to Amanita. Gosia, you also have your hand up again. Go right ahead.
Oh, you're muted.
My apologies for asking all these questions, but I just feel like
That's okay.
I'm on point. I got to be on point. So you mentioned when I found these little mushrooms all over my property, and you're right, it was a drought season. So when you mentioned drought season, Mariah, you mentioned it also. I know plants go deeper, like certain flowers go deeper, trees go deeper, and that makes them stronger. And from what I've been learning, the mushrooms are the end all of what's existing there. Like, all right, we're gonna fruit now because the cellulose is gone and the water is gonna be gone, and we got to fruit now. Is that correct?
It could be for some, yeah. Definitely being subject to stress from the environment can just make them, well, it's time to fruit with what we have now because it's not going to get any better. That's one strategy. Other might just remain dormant. The mycelium might just try to minimize any effort and wait for next year. So you might go one way or the other, and probably different fungi have different strategies. But one of the strategies, yes, it could very well be, well, let's just produce the fruiting bodies now that we have some resources. Yeah.
So this is a very heavy question for all of us right now. Are the fruiting mushrooms telling us what's lacking for the future? So if these mushrooms are starting to pop up, like, hey, we're dry. And this mushroom saying, hey, we're lacking in this. Like, it's a deep thought. It's very deep. But that's what I just felt like I needed to say. Thank you.
I don't think like you have to look at more general patterns to kind of to get a more global process or a connection between, for example, fruiting patterns of mushrooms and what might be happening globally in terms of resources or nutrients that are available, you have to do like a big study, because most of the things that you could observe, like when you were talking about the ones that were fruiting on your property, you're looking at a very specific local event. And whether that relates to something that is more global or more widespread, you would really need more data points to really make that connection. But again, with more global data, again, it would be like a completely different approach to study that. If we can look at, fruiting patterns of mushrooms and extract a conclusion about what is happening in terms of, nutrient cycling in the ecosystems or something. It could be done, but we would need so much data to be able to say something about that.
Fund mycology, everyone.
Yeah.
And actually, I felt like that you kind of bring it back full circle there, where the herbarium data, if we were going to compare, say we're talking about maybe climate change, if we're going to compare before to after, we really need to be able to go back into that type material and into also the older collections and confirm sequences on older collections. And so these herbaria are so important.
Yeah.
Gold mines for us just as much as the woods today. I just wanted to say again, because we're hitting it around past 8:30, and I think that might be bedtime for some people.
It's 10:30 in Atlantic.
10:30. Yes. So that is astounding also that you've been able to so cogently answer all of our questions. And Alfredo, I can't thank you enough for your time and your time, not just tonight, but all the work that you've done and do. So
it's great. Yeah. It doesn't take a lot to get me talking about mushrooms.
Well, you do with the best of them. So yeah, and real passion for the work. So yeah, a huge amount of love here. Yeah. And I hope you sleep well, dream of all the Pluteus, figure them all out. And we'll try to do our part here. So
okay, thank you so much.
Thank you so much, and thank you everyone for attending, and I'm going to say good night to y'all. Also, Pluteus dreams to everyone.
Yeah.
Yep. All right. Good night.
Bye.